Hello, everyone. So my name is Luisa Mejia, and I am going to talk to you about my magister thesis. In this study, well, first of all, why are we doing this? So oil palm is a really important crop here in Latin America and worldwide, and is affected by disease caused by the oomycete pathogen called Phytophthora palmivora. Oomycetes are a group of organisms similar to fungi, but really not that close related. They are most close related to algae and really closer to plants. So we have a problem, and it's that control strategies right now are more focused on good practices in the crop. And we don't have really targeted control strategies. So we want to develop them. So why do we choose metabolism? First of all, metabolism is the collection of reactions that occurs in an organism to transform nutrients into compounds of the organism, biomass compounds. And this is really important because metabolism also it's related to energy obtention. And the way that we use energy will determine the abilities of the microorganism. So with this research, we want to identify the biochemical potential of Phytophthora palmivora to find out which are the most relevant pathways in this organism. For this, we are using a tool named genome scale metabolic modeling. And this tool is based on the fact that we can predict the proteins from the genome or the DNA sequence. And we use the genome to predict proteins, those related with metabolic reactions, enzymes. And then we can reconstruct a big network that represents all the reactions that a particular organism can do. This is a general pipeline from metabolic modeling. So as we said, we took the genome sequence, we annotate the metabolic enzymes, and we reconstruct the network. This reconstruction is curated by information in literature and also from some experimental data. So these have two parts. Like one, it's to know the biochemical potential, this network, but there are another part that is to predict the use of energy or the use of mass in the organism. So for this, we do a mathematical analysis called flux balance analysis. This flux balance analysis is basically to compile the stoichiometric information of all the reactions from the network into a matrix. This kind of matrix where we have the reactions and the metabolites or compounds, and we have the stoichiometrical information, and we optimized the production of some compounds. What all this means is that if we know that biomass is like amino acids, lipids from the membrane, etc., we can optimize the production of those compounds, and we can trace which reactions are needed to get that biomass compound, those biomass compounds. So that's what we did. We took the genome of Phytophthora palmivora, and we annotate or predict the proteins, and we took those ones related with metabolism and reconstruct the metabolic network. Then for the flux balance analysis, we optimize the biomass production. This means that we took a lot of compounds previously described in literature, and we simulate the growth in a nutritive environment. Our results from the metabolic reconstruction, these are some numbers from the model. So we have that this organism have 151 metabolic pathways, bueno, a lot of enzymes, reactions, transporters, etc. These are the classes in which those pathways are related: biosynthesis, detoxification, etc. And all these talk to us about the biochemical potential of the organism. And this is important because we found some important proteins. As an example, these are all proteins related to degradation of cell wall. And degradation of cell wall, it's the first step that need the pathogen to get into the host. In this case, that is a plant host. And there are so other important pathways like degradation of sugars. This is important because sucrose, it's the production of photosynthesis. So this is the main nutrient in plants. Our FBI results, I'm going to pass quickly, but basically this means that our model is able to simulate the growth of the pathogen. So it has all the reactions needed to growth. And we find out that for energy, as an example, energy production is not mainly from glycolysis, but a glyoxylate cycle. And glyoxylate cycle, it's a metabolic pathway that allow us to produce energy from molecules like fatty acids. So all this means that we are in a first step to a better know to the pathogen, and better knowledge of the pathogen allow us to develop more efficient control strategies. And what are we gonna do? We need to fit the model with experimental data. So we are doing growth curves. We are determining biomass composition of the pathogen to improve the flux balance analysis. And this kind of model works like an onion. It has many layers. So we can fit the model with a lot of data. We can fit them with transcriptome, with proteome, or metabolomics data. And with more data we get, it's more knowledge we are gonna have. So that is our work. Thank you very much. Questions?
Thanks for the presentation. If someone has some questions, you're free to make it. No?
Thank you.
No? Oh.
This is coming. We already have some data about growth curves, but we are still in the integration of this data to the models. Yeah, it's good. Yeah.